Good afternoon, everyone. Thanks so much for joining us here today. I'd like to welcome you to NCI's MATCH Trial Update for Patient Advocates, jointly hosted by the National Cancer Institute and ECOG-ACRIN. We're thrilled to have everyone on the line joining us, as well as our speakers for today. My name is John Freimuth. I'm an Advocacy Relations Manager at NCI's Office of Advocacy Relations. Our office works with program staff here at the Institute to create and maintain a culture of advocate engagement. One of the ways we do this is by holding webinars to share information about NCI, including major initiatives and trials with the advocacy community. Today we'll be hearing from our speakers about the NCI-funded MATCH trial that is being run by ECOG-ACRIN. The MATCH trial, which stands for Molecular Analysis for Therapy Choice, is a precision medicine clinical trial launched in August of 2015. This webinar will include information on a variety of topics about the MATCH trial, including the new process for patients looking to be screened and potentially enrolled, as well as preliminary data and the ways in which advocates have contributed to the trial. Our speakers include Dr. Alice Chen, head of the Early Clinical Trials Development Program and Developmental Therapeutics Clinic at NCI, who serves as co-primary investigator for the NCI-MATCH trial. Dr. Edith Mitchell, clinical professor, Department of Medicine and Medical Oncology at the Sidney Kimmel Medical College at Thomas Jefferson University, who serves as toxicity co-chair for the NCI-MATCH trial. And Mary Lou Smith, co-founder of Research Advocacy Network, who serves as co-chair of the ECOG-ACRIN Cancer Research Group's Cancer Research Advocacy Committee, as well as the chair of the Patient Advocate Working Group for the NCI-MATCH trial. In an effort to reduce background noise, we've muted all lines on entry. We'll have a Q&A session at the end. Please be aware that this webinar is being recorded for later on-demand viewing. And participants, for the Q&A session, please submit any questions to me via the chat feature. We'll try to get to all. We might not be able to, but we will definitely make sure to follow up for any additional questions. And with that, I want to thank everyone again for being on the call, for our speakers and everyone who's helped with this effort. And I will go ahead and turn it over to Dr. Chen in just a second.
Self-mute. Okay.
There we go. Hi, Dr. Chen.
Because I saw the mute button, I wasn't sure I was coming through. So I want to thank everyone for being here and your interest in the NCI-MATCH trial. Next slide. Can you move to two slides? The slide after that. So I do want to stress, go back one. On behalf of the NCI-MATCH study leadership, and the reason I want to present this slide is that behind every one of those names, there's probably 100 people that are behind there that has put this trial together. It really took a whole village, and it's a very exciting trial for the NCI. The other part that's not on this slide is that we have pharma partners that participated with us to provide drug for this trial and to the patients. Next slide. So NCI-MATCH is part of a large precision medicine portfolio that the NCI has that is trying to use a different direction in which to treat cancer. Instead of treating it because it's breast cancer, lung cancer, we wanted to see if we find a molecular abnormality within the cancer, can we use that to determine what type of treatment may be best. Next slide. Part of this came out with the reason precision medicine, where we're tailoring treatments because we have better capability to determine inside the tumor what are particular abnormalities, and these abnormalities that potentially may be driving the cancer, and that there's a lot of drugs out there now that are able to target these abnormalities. So one good example is V600E. It's a mutation that we know drives cancer. And in melanoma, we know that if we use a BRAF inhibitor, it is a very good treatment for melanoma. On the other hand, we know that in colon cancer, even those patients with the V600E mutation, it doesn't work. So it is important to, across the board, look at different histologies, but look at that using the mutation as our similar factor when we look at how to treat and kind of decide where the best patient population may be. Next slide, please. So NCI-MATCH is designed to look at if we can find these specific mutations, or what we call actionable mutations in tumor, and then use that to target the treatment that we want to use, can we have better success in managing the cancer? This is a signal finding trial in the sense that we want to use this trial to get leads and to further expand in other trials to better define what may be the right population, the right timing to use these drugs. Next slide. The design of NCI-MATCH itself is unique as well in that we have a master protocol. The large overarching protocol that says the patient has to have good kidney function, certain performance status, certain similar features. But then once we find their actionable mutation, they go into one of the treatment arms, and there are 35 treatment arms through the lifetime of NCI-MATCH. And we actually have 4 additional arms that by late summer 2019 will open. So NCI-MATCH will end up having 39 arms. Some of the arms now have actually closed. 10 arms has been reported or will be reported soon. But basically, the tumor gene testing will help us direct the patient to a specific arm, not the histology itself. Next slide. This is an adult trial. Because adults and children have different types of tumors and different actionable mutations behind those tumors, there is a pediatric match trial that looks at more pediatric-specific tumors. It is in solid tumor, lymphoma, and myeloma patients. Patients have to have at least progressed on 1 line of standard care, or if there's more than 1 line that actually known to prolong overall survival, because that's already a known improvement in the survival, they would have had to have that prior to coming on to NCI-MATCH. So it doesn't exclude them, they just have to have seen that treatment previously, or that we know they can't get it for medical issues. Next slide. In order for us to capture what we think potentially may be some very rare driver's mutation, we need to have a lot of different sites. And through the NCTN, or National Clinical Trial Network of NCI, as well as NCORP, which is our community oncology sites, there are 1,100 sites that are open and participating in NCI-MATCH, including District of Columbia and Puerto Rico, and every state, as well as DC and Puerto Rico, have put 1 patient onto the NCI-MATCH trial. The next slide. This is where the NCI-MATCH location is. Next slide. The referral pattern for NCI-MATCH is that patients need to come in with a known mutation for one of the arms that are open. So they have to have lab identify that mutation. The current practice for oncology now for a lot of cancer patients is that they will get these mutational analysis done because there are so many treatments out there that is dependent on this. So as a routine care, the physician will order this. And for one of the labs that NCI has worked with, who is part of the NCI-MATCH referring labs, they will then work to identify these patients for us. So NCI has worked with various commercial and academic labs to validate assays to the NCI-MATCH assays so that we know that pretty much the results are going to be the same regardless of which lab does it. This allows us to continue to use the 1,100 sites to accrue patients, and these labs are readily available at these sites so that it allows us then to still allow patients to have access to this trial close to home. Next slide. So this is more a pictorial pathway, and I'll go through each one. Physician sees a patient, and they send the patient gets biopsy, the tumor is sent for genomic testing as routine care, and not specifically for NCI-MATCH. The lab runs the standard test, send the result back to the physician, Now, if it's an NCI-MATCH site that identifies the patient and the lab is an NCI-MATCH referral laboratory, the lab will look at that result to see if the patient has an actionable mutation in one of the open arms for match. If they see that, they will send the referring physician a letter or an email notification that, dear doctor, your patient potentially may be eligible for an arm for NCI-MATCH. That allows the patient and the physician to decide if they want to enroll onto NCI-MATCH. If they do, the physician at the NCI-MATCH site will then enroll the patient onto the screening or master protocol. This allows the lab to get a notification that they should upload the patient's info into the match database. Once the lab data is uploaded into the system, the trial leaders will get those information, and we meet every day. We will look at this and make sure that the mutation is correct and no error has occurred, that this is not a histology that's specifically excluded for an arm. So even though I mentioned that their histology is not used to identify patients for any particular arm, we do exclude certain histologies in each arm, either because we already know the drug works for that histology or we know the drug doesn't work. Like colon cancer for BRAF is excluded because we know it doesn't work, or that melanoma is excluded because it's already approved for that indication. We want to use those 35 patients per arm to help us explore areas that we don't know the answer to yet. So once the trial leaders assign and verify patients to arm X, and that notification is sent to the physician then will reevaluate the patient to make sure that they're still eligible for arm X. Their kidney functions are okay, their liver functions okay, patient's able to swallow pills, things like that. If patient is eligible, they will then enroll the patient onto arm X for a treatment, and drug will be sent to the site, and patient will start treatment for match. Next slide. There are 10 commercial labs currently that has gone through the process of validation that can make general referrals to NCI-MATCH. Next slide. There are additionally 14 academic labs that will refer patients at their own site. So it's at MD Anderson, somebody is seen there as a consult. They send the specimen to Anderson, does their own analysis, and it looks like they're eligible for arm X. Then they will go through the same process. Next slide. This is a list of, or this is the website for looking at the latest labs that are open for NCI-MATCH referral. Next slide. I want to acknowledge the advocates that are involved in the trial design and help us to oversee the conduct of this trial. There is widespread participation and, as I mentioned, the whole village, but mostly I want to thank the patients and the participating sites, all the local sites that are very excited and were able to refer patients and consider them for NCI-MATCH. So I think that is my last slide.
Yes, thank you so much, Dr. Chen. And if everyone could bear with me for just a moment while I pass the presenter ball over to Dr. Mitchell so she can share her presentation. Hi, Dr. Mitchell. Have you received the presenter ball yet? Hi, Dr. Mitchell. You might be on mute. Could you unmute your line, please? Hi, Dr. Mitchell. I think you might be on mute. Could you unmute your line, please? Okay, thank you. Dr. Mitchell, does this work now?
I can hear you. You hear me?
Okay, wonderful. We can hear you now. If everyone else could mute their line, please, just to avoid background noise. Thank you so much. Apologies for that hiccup there.
And John, I don't see your ball, so go on and advance.
Oh, okay, we'll do.
That's not my dog barking.
If everyone could mute their lines, please, other than Dr. Mitchell, that would be very helpful. Thank you so much.
So I will discuss the individual treatment arms and the trial demographics and how trial proceeds and some of our results. Next slide, please. So of the patients enrolled for treatment, there have been two processes. One is the current laboratory process where the laboratories refer patients, and the previous was the central screening process. Of the current referrals, there have been 246 patients admitted to the MATCH trial, and for the previous process of central screening, 689 patients for a total of 935 patients admitted to the MATCH trial and have been treated on the trial. The average weekly enrollment is now at 7 patients per week. The percentage of patients who receive a laboratory referral high that receive treatment. All of the patients who received a laboratory referral, 81% of those patients have received treatment. So therefore, enrollment in the trial follows through a process, and the chances of a patient's tumor having a genomic profile that matches one of the arms of the MATCH trial is very high. Next slide, please. Of the 39 arms of this trial, not all arms have opened at the same time. And if you will notice on your screen to the left in gray, there are 4 new trials that are in development. The green ball means that 16 arms are currently open to enrollment at this time. In yellow, 3 arms that have met enrollment accrual and therefore admission to those arms suspended. In red, there are 6 arms that have completed accrual and are in follow-up. And at the top arrow, there are 10 arms that have completed accrual, and we are now analyzing the results of those, with some being already reported. Next slide, please. So the overall design of the trials allows for, once a patient is enrolled on a treatment arm, those patients will be treated for as long as their tumors continue to shrink or respond, or if the tumors remain stable on imaging and other evaluation processes. So therefore, if one looks at the pink arrow, that once a patient enrolls on an arm, there is continuous evaluation with the treating physician or center. And if there is a complete response, partial response, or stable disease, they can continue on study unless there is progressive disease or if the patient is intolerant far as having problems or toxicities. And we've actually had a number of patients who have been on study receiving treatment for more than a year. So there have been some responses, and we are very appreciative both for the patient and their treating center for the follow-up. Next slide, please. Once there is evidence of disease progression, to aid in the research and to determine if there has been a change in the tumor, a change in the genomic profile or markers, we would like to know that. And consequently, we do request that if there is evidence of disease progression on the trial, that a repeat biopsy is accomplished and that biopsy is sent to us so that we can continue the research effort in understanding genomic profiling and response to drugs based on a match of the genomic profile of the tumor. Next slide, please. So for the objectives of this trial, the primary objective and research goal is to understand the proportion of patients with refractory either solid tumors, lymphomas, or multiple myeloma who have experienced an objective response while on the therapy. And if the overall response rate is greater than or equal to 5 to 31 patients, or 16%, it was determined that this particular agent may be worthy of further study. Secondary objectives include progression-free survival at 6 months, time to progression and or death or other problems, the toxicities experienced by patients, and potential predictive biomarkers that will allow us to select or better select therapy. Next slide, please. So the goal of each of the 39 arms is to reach accrual of 35. Some of the studies have been past 30 patients, but our goal is 35 on each arm, and a total of 31 patients evaluable for the protocol. Next slide, please. So of the arms of the study that have already reached accrual and our accrual goal, note that we have actually response rates already determined in several of the studies. And note in yellow, this is a busy slide, but the important information is in the yellow marks. So notice that for some of the trials, there has been all responses, 8%, trial, 0%. Note at the lower level of this yellow area that there is one trial that has accomplished 23%.
According to Wikipedia,
So therefore, this is very important. And we're just so excited about the evaluation process that we're going through now in the trials and the arms of the study that have already completed accrual. Next slide, please. Next slide. Thank you. As we evaluate all of the patients that have either been assigned treatment or enrolled in one of the arms, we want to understand who are the patients and which sites have accrued the patients so that we have access throughout the United States and other areas of participation in the MATCH trial. So note that the majority of participants who have been assigned therapy was 60% female, and of those enrolled on the one of the 39 arms has been 62% female participants. Next slide, please. Of those patients who have been either assigned therapy or enrolled on the arms, it is very important that we understand the background of the patients, and we also do know their locations throughout the country for accrual. But note that 81% of the patients who were assigned therapy on the arms were white, and of those enrolled on the arms, 83.4% white. Note that a few patients may have changed their treatment plans or for other reasons were assigned therapy but never enrolled. So the enrollment is different from those who have completed the admission process and who were assigned therapy. 8.9% African American or blacks assigned, and 7.6% African American or blacks enrolled on the arms. These were the two major components of those admitted to the trials, and note that there are additional percentages identified for those individuals who identified as Asian, Native Hawaiian, Native American, multi-race, or their race was not reported.
Next slide.
Cross-section individuals on the trial. According to ethnicity, the assigned arms and enrolled on arms are given as Hispanic, 5.6% assigned, not Hispanic, 90.2%, and unknown ethnicity, 4.2%. So in summary, understanding the patients enrolled on the trials, who received therapy on the trials, and the genomic profile of the tumors that these patients experienced is really key to our understanding of the NCI-MATCH trial. And I'd like to personally thank, in addition to those who have helped organize and manage this trial, I want to thank the patients and their treating clinicians and their treatment teams in helping us collect all of the information and the data in relationship to the MATCH trial. Thank you very much.
Thank you so much, Dr. Mitchell. We really appreciate it. And now we will be turning it over to Mary Lou Smith, who will be discussing advocate involvement.
Hello.
Hi, we can hear you, Mary Lou.
Okay, great. Thanks. Good afternoon, everyone. It's my pleasure to talk with you today about how advocates were and continue to be involved in this trial. Next slide, please. So the Patient Advocate Working Group was established in 2013 at the request of ECOG-ACRIN. It was and still is composed of advocate representatives from each of the NCTN groups and the National Cancer Institute. As advocates, we always recommend bringing the patient voice to trial design early in the process, when design elements may be changed to reflect patient needs and concerns. And that's exactly what the NCI-MATCH principal investigators did. And two things they especially wanted help with was the mandatory biopsy and the drug holiday. To respond to their request, we sought input from other advocates and the physicians, nurses, and staff from ECOG-ACRIN. The PIs listened to what we had to say and adjusted the design based on patient preferences. They asked us to review and comment on the informed consent document. We suggested language changes, obviously, and recommended the inclusion of the description and rationale for the mandatory biopsy, thinking it would be helpful to potential participants in making an informed decision. We interviewed patients who had registered to Step 0 of the MATCH trial about the issues they had encountered. These issues included time off treatment, expectations of having a mutation or alteration that matched to an arm of the trial, and what options they had if they did not match. We shared these issues with ECOG-ACRIN at their semi-annual meeting. Next slide, please. The MATCH researchers asked us to provide information on other patient issues with precision medicine trials that would help them in the design of the follow-up trial. ECOG-ACRIN and Research Advocacy Network received a PCORI grant to conduct a precision medicine symposium in 2017. All of the presentations and slides from that symposium are available on the Research Advocacy Network website. The invitees included the advocates from the NCTN groups and the members of the National Cancer Institute Patient Advocate Steering Committee. The symposium highlighted the MATCH trial. We held a think tank the second day to identify patient issues with precision medicine trials. The issues identified came under the broad headings of communication, logistics, and clinical trial design. We recently conducted a follow-up survey to validate that these issues are still relevant. There was unanimous agreement that they are. There were also some additional issues thought to be critical by these respondents. For example, access, education, and health literacy. Our survey asked for initiatives that the respondents were aware of that could address the identified issues. The projects they mentioned included the ASCO and Friends of Cancer Research Project on broadening eligibility criteria, the Multiple Myeloma Research Foundation's My Drug, PanCan's Precision Promise, Longevity's Take Aim initiative, and SWOG's Plain Language Project. A complete report on the survey results will be available on our website by the end of March, and we'll be holding a web event and an online group to discuss the report and next steps to encourage solutions to all these problems that we have identified. So thank you very much.
Fantastic. Thank you so much, Mary Lou. We really appreciate it. Thank you, everyone, for joining, and for those who submitted a question. We've received just a few via the chat feature. Again, if you have any others, please feel free to submit them that way. I'd like to start off with the first one we received, asking if any of the speakers would be willing to share what efforts were made to recruit from community settings such as public hospitals that might serve more diverse racial and ethnic populations. What efforts were made to connect with those communities and recruit potential participants from those communities?
So I can answer that. This is Edith Mitchell. With ECOG-ACRIN, we have had several meetings prior to the initiation of patient accrual to various National Cancer Institute programs. The ECOG-ACRIN program, which occurs twice per year, we've met with not only physicians, but nurses and research assistants from sites throughout the United States, including community practices, community centers, and community hospitals. In addition to ECOG-ACRIN meetings, we spoke at other NCI meetings, such as the Alliance, SWOG, and other meetings. Additionally, we had meetings in various cities so that we could recruit equity in the patient population throughout the United States. So, yes, we met with patient advocacy groups, hospital groups, National Cancer Institute programs, and others so that we could, one, engender interest in the trial and making sure that we had information to various sites throughout the country. So, in summary, a major effort was made to give out information about the trial, to describe the accrual method and the steps that would occur in terms of meeting the requirements of the protocol. So big efforts throughout the country with many individuals involved.
Fantastic. Thank you so much, Dr. Mitchell. Another question we received is, what is the next steps for drugs that have been found to be effective?
So this is House Chen. We have, with each arm, as they mature, have looked at the results, and there are further meetings based on those results to see if there are interesting leads and where additionally to take those leads, starting with meeting with CTAP and meeting with the pharmaceutical company. There is also additional samples that were collected from the first 6,000 patients and archival tissues that are being requested, to which we are going to analyze those tissues further for additional actionable mutations, looking for additional abnormalities that may help explain either response or lack of response. And with those information as they become available, if there are interesting leads, again, there will be, we are looking at those. We're having regular meetings to see if these should be taken further into clinical trials. With each of the arms, there are young investigators that are experts in these areas, both as clinicians as well as translational scientists, so that they can help us define these leads, see if they're interesting, see where the field is going, see if there are additional areas we need to take. Like for our one of our Z1D arm had interesting results, and so we are in the process of opening another arm, taking that one step further and doing a combination trial with it. So it's a very dynamic trial process in terms of constantly we're looking at the data, looking at what the whole precision medicine field is, and looking at that specific drug and similar drugs in that area to see if there are changes we need to make, new mutations we need to add for trial arms that are open. So, it's we're wanting to make use of every lead that comes out of this trial.
Thank you so much, Dr. Chen. And I think another question is, if, let's just say, for instance, I'm a patient and I have my sequencing in hand, how would I go about enrolling? What would be the first step?
So, hi, I can answer that. If a patient already has their tumor profile, what they should do first is to discuss with their physician or treatment team about the profile that they have, evaluate which laboratory and whether that laboratory that performed the test is on our list of laboratory networks. So make sure that that laboratory is in the network. And then the patient's physician or treatment team can contact either ECOG-ACRIN to move forward, or contact that laboratory. So the laboratory can provide a letter to the physician saying that this is eligible for the MATCH study, and then we move ahead with all of the information for accrual. But the patient's physician and treatment staff must be involved early on, and they are the ones to submit the information, and then once there is accrual to the study, if that results in accrual, they will have to administer the medication. So the treatment staff must be intimately involved at all stages.
Fantastic. Thank you so much. And another question is, how can advocates and advocacy organizations help connect patients with the MATCH trial for potentially informational purposes, or if they're just interested in potentially enrolling?
So, John, I can answer that also. For one thing that the advocates and caretakers and others can do, there is something that all of us can do to support the MATCH trial. One is to utilize the website so that we have more individuals who know about the trial, discuss it with potential patients or other family members who might be involved, and discuss it with their treatment team, with their physicians and nurses, so that more people are involved in the MATCH trial and the evaluation and accrual of patients. So there's something here for everyone. I am so delighted and appreciative of the advocates and supporting individuals and others who are helping us with this trial. And thanks for attending the webinar today.
Wonderful. Thank you, Dr. Oh, I'm sorry. Go ahead.
I was just going to say one of the things that, just understanding the process of the referral, it is a little more complex than in general, because it does require that patients have that test result, have that genomic testing done already that identifies them as having a potential actionable mutation. So kind of where that process and how to get into the system, I think understanding that process will help patients and patient advocates in terms of referring patients for NCI-MATCH.
Thank you so much, Dr. Chen. I think we have time for a few more questions. One person was asking if, I hope I'm conveying this correctly, so further studies indicated if overall response rate is more than 5 patients, they were wondering if, at the same time, if there was a truly exceptional response in just one patient on an arm who had long-term stable disease where no one else did. Are there any plans to investigate cases like that to learn from that patient or what might be going on?
Yes. So the response rates, looking over the whole arm, is kind of to help us direct kind of future directions. But I think each individual patient within that arm still in itself is an indicator to help us understand the drug and that mutation better. So in one particular arm, we have several rare tumors that shows that there may be activity, even though it's only 2 patients. It is a rare tumor, and we felt like that is area probably will deserve further evaluation of that particular actionable mutation in that histology. As I mentioned, we are going to do additional genomic testing on the back end, and especially the exceptional responders will be of great interest to us. And even, or just as important, is to understand those patients that responded and then progressed, if we can get biopsies to help us understand what was the resistant mechanism that occurred that caused the progression. So yes, every individual patient in this trial is very important to us so that it helps us understand how the drug works, the response. Every patient in the trial may not just have one actionable mutation. They may have other genomic characteristics that either help define their response or define their resistance. So we are looking at the whole, but we're also looking at the individual to help us understand better what is going on.
Oh, certainly. And also, for each individual patient, we are evaluating the potential toxicities and how the drugs for the MATCH study might interfere with other medications that the patient is on. So every single patient on this match study is evaluated carefully, both in terms of treatment response, potential toxicities, trial, and whatever we can find out regarding responses and responses followed by tumor progression. Every single patient is very important and totally evaluated so that we can find out more about how we can incorporate precision medicine into overall cancer treatment planning.
Thank you so much. We're still getting quite a few questions in here. Someone wrote, with all the effort that has been made to include a more racially diverse patient population, have the percentages improved over time? And also kind of another follow-up question, if the study team was surprised with the amount of diversity that was that's there given the number of sites for the MATCH trial.
So this is Edith Mitchell. The percentages of the various populations that I described has changed with time, and that is due to a number of reasons. One, the sites that were participating at the time. Also, the markers, some of the genomic markers tend to be more frequently seen in various populations, as well as the tumors that were prevalent in some of the arms. So this has changed and actually changes consistently from week to week. And we're evaluating that in a total manner, but yes, there have been changes dependent on a number of factors, including whether the cancer or the disease process was more commonly seen in certain populations. So we're evaluating all of that, as well as factors related to which patients might agree to going on a trial such as the NCI-MATCH trial.
Fantastic. Thank you so much. And I think it gets into a little bit of one of the other questions mentioned, which is, if I'm understanding correctly, since about 19% of patients who were originally thought to be appropriate for one arm or another and might not have been, what was the analysis of the rationale for that, for a patient not being matched? For those who aren't matched to a particular, what are the options for those patient groups to work with their physician?
So we really work with the clinical sites that submit patients. So we try to find out as much as possible. For some of the arms, for example, if there is a drug already approved for a specific cancer, we would not want individuals who have that cancer to be on the MATCH trial because data are already available. But we would want patients who have different tumors. For example, if there is in one of the arms, the drugs were against BRCA. There was a breast cancer patient. They were usually and we were looking for other tumors whose tumors may express BRCA or other markers. So we worked very carefully to increase the opportunities for patients, especially if their tumors carried markers that are universally thought to be typical of other cancers.
you so much. I think we will, that is actually all the questions I've received via the chat feature, I believe. So if anyone else, maybe we'll give another 10 or 15 seconds for anyone else to submit one. And otherwise, I think we can break a little bit early. I want to, again, thank everyone so much for joining. I hope this information has been helpful. I, of course, want to thank the speakers so much for all their work and really everyone who was involved in putting this webinar on. We truly appreciate it and are very excited to share this sort of information with the advocacy community. I am not receiving any more questions via the chat feature, so unless I'll turn it back over to the speakers in case they'd like to add something as a final note, but otherwise I think we can conclude the webinar here.
So this is Al Chen. I just want to thank the advocacy group for all their interest in this trial. The success of this trial, I think, depends heavily on the patients, the advocacy group, you know, and a lot of investigators in terms of their enthusiasm, and the physicians for this trial. And I think the goals and achieving some of the goals we achieved so far is largely due to the enthusiasm for it. Thank you.
John, I'd like to say ditto there. We are so appreciative to everyone that has contributed to the success of the NCI-MATCH trial. Thank you, advocates, caretakers, patients, clinical sites, and all involved with the trial. It does continue, and we certainly would like to have everyone to continue work with us if there are questions. We have multiple sites for questions, and we are just so appreciative of the patients, families, their caretakers, and the patient advocacy groups that have contributed immensely to this project.
And this is Mary Lou. I'd like to know if there's anything that the advocates need around the MATCH trial. Is there any information? As time goes on, please contact us.
Absolutely, and I put some resources we have on that last slide there up for everyone to see. And again, I'd just like to thank everyone so much for joining us here today, and we really appreciate, again, you guys tuning in and everyone's work on the project, the MATCH trial, as well as the webinar here itself. And if anyone has any questions, again, as Mary Lou said, please feel free. You probably received this invite through our main inbox, so feel free to shoot us an email. We don't want anyone to think this is the last chance to ask questions about the trial. Our door is certainly always open. So with that, I will thank everyone again, and we will go ahead and end the meeting here.
Thank you, John.
Take care, everyone. Have a wonderful afternoon.
Wow. Definitely worth listening to.